Hello, AP Bio. Today we are going to learn about linked genes and another mechanism of genetic inheritance. So, to get us started today, I want you to think about this set of circumstances. You need to test a gene's inheritance pattern. And in order to test this gene's inheritance pattern, you're going to need an organism. You're imagining that you're inserting this gene into an organism that you can observe inheritance in, because you can only observe inheritance in multiple generations. You need an organism that reproduces really fast. You need lots of offspring, and you need simple genomes to avoid conflicting gene interactions. So, based on this information, I want you to think about what kind of organism you might choose. Hopefully, you had a chance to think about it. If you haven't, definitely pause the video. If you said any kind of insect, then I would say you're very close. You're very close to what you would be looking at. Now, if you're in my class, these are the documents that you're going to need in order to understand this lesson. Okay, so it's the Thomas Morgan Fruit Fly Genetics Experiment Analysis. And then this is the image that we are going to use for this particular lesson, this first part of this lesson. So make sure that you have these. If you are in my class, I also strongly recommend that you highlight or color this diagram because it was printed in black and white, and it's really helpful to see it in color. And so I really strongly recommend you go back and color this diagram based on what you see here in order to best understand this lesson. So Thomas Hunt Morgan had the same question that we originally started with. What organism should we choose? He chose fruit flies. Fruit flies produce huge numbers of offspring. New generations can breed every 2 weeks, and they only have 4 chromosomes. This is one of the reasons that I am not a geneticist, because I do not want to work with fruit flies. Because if you are a geneticist today, you still work with fruit flies to understand how genetics works and how inheritance occurs in a bunch of different genes. So this is what these flies actually look like. They are gross and nasty, but they are really, really effective for observing the inheritance of certain traits and genetic phenomena. Now, there's some terminology that you need to know in order to understand what he did with these experiments. So the wild type terminology refers to what the non-mutant type would be. This is basically the average run-of-the-mill everyday fruit fly. Okay. We're specifically looking at eye color right now. We're going to focus on eye color. And as you can see, there's a variety of different types of eye colors. All of the variations of eye color besides the red eye color are what would be considered mutant phenotypes. So you can have white fruit fly eyes, which are these ones right here. You can have cinnabar, which is this sort of like cinnamony color. You can have black eyes. There's a bunch of variations that you can have in the eyes of fruit flies. Now, a couple of other pieces of terminology you're going to need. The parental type is the offspring with the same phenotype as one of the parents. Okay, so the parental type is the offspring with the same phenotype as one of the parents. The recombinant type is offspring with a different phenotype than either of the parents have. So let's take a look at a specific trait. Let's look at eye color. In fruit flies, red is dominant to mutant types. Eye color is also sex-linked. So which of the offspring genotypes below is or are, if there are multiple, the recombinant phenotype? So looking at this Punnett square, if you see the plus symbol indicates that it is the wild type. Okay, it is the red wild type. If it doesn't have the plus symbol, that means it is the mutant type. So see if you can answer this question. Which of these would be the recombinant phenotype? Did you get it right? So looking at these recombinant phenotypes, the reason these are considered recombinant is because they are not like either of the parental groups. This would be a mutant female, and this female is a wild type female, and this would be a wild type male, whereas the parent was a mutant male. So let's use some of this terminology. Again, that diagram that's in your packet is going to be essential for understanding and going through these questions. I want you to try to answer these first three questions on your document. What are the traits Morgan was observing in this cross? What were those traits represented by? And what phenotypes were in the parent generation? Then what is their genotype? Finally, what types were observed in the F1 offspring? What phenotypes were observed? And what was their phenotypic ratio? Remember, in fruit flies, red is dominant to mutant, and eye color is also sex-linked. So take a second and see if you can answer these questions. I would give yourself at least 5 to 10 minutes to answer those three questions. Definitely pause the video at this point too. So this is the part of the diagram that we want to be paying attention to. Thomas Hunt Morgan originally took in the parental generation two homozygous individuals. And you can see here, he is mapping the trait for body color and wing shape. So that's what we're going to be mapping, okay? He's looking at the gray body versus black body type and the normal wing versus vestigial wing type. And if you look here, vestigial wings is going to be this sort of like gimpy looking wing structure. So he bred those two together and he bred homozygous individuals. Now, in this case, we are using plus to represent the wild type, and the wild type is dominant to the mutant type. So if you look at the next generation only produced one type of offspring. And I want you to try to see if you can figure out why it only produced one type of offspring. So these are your alleles. You have the B plus, which is for the gray body, the VG plus, which is for the normal wings. Black body is the B, and vestigial wings is the VG. What phenotypes were observed in the parent generation? Okay, so we went over this. We went over the phenotypes and the genotypes. Okay. Now, what types were observed in the F1 offspring, and what was their phenotypic ratio? Well, if you look at this dihybrid cross Punnett square, you don't have to do the dihybrid cross. I gave it to you just right here. I want you to try to see if you can figure out what the ratio would be of the phenotypes. And again, look at this part of the diagram. What would be the ratio of phenotypes that occurred in the F1 generation? Did you get 1 to 0? Because 1 to 0 would be the correct answer. 16 out of 16 possible genetic combinations gave you all the same possible offspring. And so the ratio would be 1 to 0 for the other genotypes. 1 to 0 to 0 to 0, because there are technically 4 different possible phenotypes. Okay. So here's another set of questions. Again, we are going through Thomas Hunt Morgan's experiment to try to decipher what it was that he did and what he discovered by following fruit fly generations. So your next set of questions: what genotypes did Morgan cross to get the F2 generation? What would be the expected ratio of phenotypic results in this cross? And what was the observed ratio of phenotypic results? Again, this is mostly analyzing the diagram, but there is going to be some math that you'll need to do. So take a look at this diagram. Pause the video and make sure you're trying to answer all these three questions. So after you've paused the video, what variations did Morgan cross to get the F2 generation? Well, if we go back, Morgan crossed one of the heterozygous females, this gray-bodied, normal-winged female, and a mutant male that had the black body and the vestigial wings. So there was a wild type from the F1 generation, gray body, normal wings, and heterozygous for those traits. And then a double mutant, meaning that it is mutant for two traits, black body and vestigial wings. And you can see that here in this top section, okay? What would be the expected ratio of phenotypic results in this cross? Well, if you do the dihybrid cross, which the dihybrid cross, the Punnett square, is how you figure out expected ratios. If you did this Punnett square, you would need to have come up with this result. You should have a 1:1:1:1 ratio of gray body, normal winged flies, gray body, vestigial winged flies, black body and normal winged flies, and black body, vestigial winged flies. I've highlighted the different genotypes, or the different phenotypes, I should say, here. So this is what your expected value should be. You take your total number of offspring. You have 2,300 total offspring. And you take that value and you multiply it by the ratios that you had. Now, if we go back and we were to think back on how we looked at the ratios for these questions, technically 1:1:1:1 ratio was a reduction. Originally, this would have been 4 out of 16, 4 out of 16, 4 out of 16, and again 4 out of 16. So if we take that ratio and apply it to the 2,300 flies, we get 571 expected gray normal flies. So again, if you're struggling with how to do this, I really recommend going back and reviewing how to find expected results, okay? We're taking our total number of offspring and multiplying it by the ratio we would expect to see, which in this case is 4 out of 16. Our observed was 965, 206, 185, and then 944. So this already should be setting off alarm bells that says this does not look like it's very close to what we would have expected to see. So how do these observed values compare to the expected values? Well, if you've been in AP Bio, you know that this is where you would do a chi-squared analysis. Because this is how you can determine if your expected ratios were close enough to your observed ratios. Just a reminder, I got these observed values from this chart. I got them from this diagram. The last generation, I got 965 wild type offspring, 944 black vestigial, 206 gray vestigial, and 185 black normal. So that's where I got these values. Okay. So this is the point where you would do a chi-squared test. Make sure if you're going to do a chi-squared test, you form a null hypothesis, you input your values into the equation, and you determine if the critical value is lower or higher than the chi-squared value. Now, our null hypothesis would be that there is no statistically significant difference between the observed and the expected results. Our chi-squared values, these are the things that you need to calculate, and this is how you would determine if you fail to reject the null or if you reject the null. So, do we reject our null hypothesis? Well, Thomas Hunt Morgan would have concluded that these offspring do not match the expected offspring. Now, I'm not going to go through the whole chi-squared calculation because that's for a separate lesson, but I encourage you to go back and do the chi-squared analysis on your own. Since these traits, these observed and expected traits, were not close enough together based on the chi-squared analysis, this led Morgan to conclude that sometimes parental traits are more likely to be found together because they are on the same chromosome. This is the concept of linked genes, the idea that they may be closer together because they are on the same chromosome. And this leads us to a question. Are the parental alleles always going to be found together? And how do you know? Well, if you were to look at this experiment and you were to look at the experiment he did, you would notice that parental alleles are not always going to be found together. If we go back to this diagram, the parental traits didn't always get associated together. Remember, the parental was the wild type versus the mutant. And if you notice, you have a mixing of the wild type and the mutant here with the gray vestigial and the black normal. Now, if you have learned about meiosis in AP biology, you will know that this is the result of crossing over. Something had to be breaking the bond between the parental traits, and that thing is crossing over. So I want you to look at these three options. Based on these three options, which of these genes do you think would most likely be linked? A, B, or C? I'm hoping that you said A, because this is what Thomas Morgan concluded. Parental traits are more likely to be found together because they are on the same chromosome. Now, if you said C, that also could have been a viable answer. But I want you to look at the distance between gene 1 and gene 2 on chromosome C. They are significantly farther apart. And so I want you to consider, okay, would A or C be more likely to undergo crossing over? So let's take a look. Linked genes are not just found on the same chromosome. They are also found near each other on the chromosome. They tend to be found together. So these are very common traits that you are probably familiar with in your daily life. Red hair and green eyes is found close together on the same chromosome. So there's often a lot of redheads who have green eyes or hazel eyes. This is true for blonde hair and blue eyes as well. However, they can be separated, and this is how you can get black-haired individuals with blue eyes via crossing over. So I'm going to stop this lesson there. I want you to go back to the Thomas Morgan fruit fly experiment analysis and see if you were able to answer all of these questions. There will be a part 2, and that part 2 will go over the rest of the understanding of linked genes, and it'll go over this guy, which is Alfred H. Sturtevant, and what he did with Morgan's data.